Hello everyone, welcome to my first and brand new channel, Botanopedia. This channel will help you know about different aspects of botany. Now, the topic today that we are going to learn is about a disease whose name is brown spot disease of rice. Now, this disease, first of all, we must know that every disease has its own synonym, which every disease may be known by other various names, which is more easier to understand just because why the name has been given to it. So this disease, brown spot disease of rice, also has a number of synonyms, which may be also referred to as the sesame spot disease. Sesame spot. Why? Because the spots on the sesame plant caused by the same disease is similar to the spots present on the rice. Now, another name is helminthosporiosis. So why is this name given? Why is this synonym given to this brown spot disease of rice? There's a reason, just because the causal organism of this disease, which is also known as Helminthosporium oryzae or Dexclera oryzae, which was the previous name given to it. So, because the disease caused is the Helminthosporium oryzae is the causal organism, so the disease is also named after the causal organism. Causal organism, which I already said, it's Helmintho. The scientific name of this causal organism is Helminthosporium oryzae. Now, and the last, which is a very common name, also known as the leaf blight. The leaf blight disease of rice. This because it affects the leaves of the plant. Plants are mostly affected. Now, after teaching you about the disease in brief, it is actually important for me to show you a pictorial display about how the disease would look like in real life if you need to identify it in the field. So first of all, let's see about the inflorescence of the rice plant. Now I said you that the inflorescence of this rice plant is known as the panicle, and the panicle looks like this. Now, if the inflorescence becomes infected in an early stage, it does not develop any grain, as because you can see here the grains have already developed. Now, depending upon the degree of damage caused by this pathogen, can cause complete sterility of the seeds or partial filling up of the grain. Now, as you can see, there are dark spots seen. These dark brown spots are seen in the glooms, and when the endosperm of this becomes brown, and that reduces the quality of the grain. So this is how the disease infects the inflorescence of the plant. Now going to the leaf sheet, you can see the leaf sheet and the leaf blade have symptoms which are quite similar. Like, see in the leaf sheet, we can see discrete dark brown elliptical eye-shaped spots. You can see eye-shaped elliptical spots. Now a fully developed spot, you can see many of them are greyish brown, having a black greyish brown center surrounded by reddish brown margin. Now these are fully developed spots which have a very fresh inoculum of the pathogen. Some are even yellowish halo which show that they are an early stage of development of the infection inside this plant. This is how it looks on the leaves. And lastly, the grains which after the harvest of these grains from the plant, you can see these are the infected grains. If the grains are infected with the infection, now see, these grains can fully depict that these are infected because these are not what normal rice grains look like, normal healthy produced rice grains. These rice grains, as you can see, they are having dark brown, rotten kind of looking. The structure kind of looks like they're rotten from inside, and they're like half burnt. The black red down spots are those inoculum of the pathogen. If these are dissected and seen inside the microscope, it would probably showing that there are well developed hyphae of this pathogen spread all over the seeds. And if the seeds are now by chance being sown and the farmer does not separate them before sowing in the soil, this may result in a development of weak and infected seedlings which grow up to spread the infection to the later stages of the plant, and those plants may even bear rice grains with infected rice grains, and this goes on and on if the mistake is neglected by the farmer. So this was a pictorial representation of. Now coming back, the second factor of a disease, the causal organism. Now the causal organism is Helminthosporium oryzae. Helminthosporium oryzae. Everyone should be careful about the spelling when you're writing about the spelling of the scientific name of this pathogen. Or it has a synonym, which is the current name given to it. It's Dexclera oryzae. Now we know this pathogen is a type of fungus. It's a fungus, and as a fungus has two stages. Imperfect or an anamorph, or a sexual stage. A sexual stage refers to a stage which actually it adopts when the conditions are unfavorable for growth. The sexual stage, which we know is this Helminthosporium oryzae. This is the asexual stage of this pathogen. And the imperfect stage, the perfect stage, or the teleomorph stage, or the sexual stage, is named as Cochleobolus miabeanus. Mind about the spelling, Cochleobolus miabeanus. Now we know that any perfect or asexual stage of any fungi, any other animal or any other organism, for producing through sexual reproduction, has to produce two types of spores. Which then fuse to form a dikaryotic mycelium. For any type of Basidiomycotina or Ascomycotina fungi, mycelium, which then grows into a fruit body. Now let's look about the causal organism. I'll be helping you know about organism in detail by a pictorial demonstration. Let's have a look. Now, I would be going through a pictorial demonstration about the causal organism Dexclera rhiza. Now, as I said this is a fungus. Now, the fungus is known to be septate, means it has divisions. As you can see in the picture shown here, they are segmented. The conidioes and the conidiophores also are segmented, and they're prostrate. You can see that they are standing erect, they're prostrate and much branched. The hyphae is much branched and they bear conidia. So the conidiophores are prostrate, septate and much branched hyphae. Now, let's look at these are the conidias. You can see the conidias are also segmented. This is a more clearer look of the segmented conidias. Now see, the conidias are segmented, they're cylindrical to ellipsoid with slightly curved, and as you can see on the picture in the microscope, it's shown that they're light brown in color. And I must also say that, see, not every conidia has tapering ends, but a common characteristics of all the conidia is like they have tapering ends on both the sides, and these both apical regions are the regions from which they germinate and grow into a new fruit body. So, as because both the tapering on the apical ends of this conidia, they germinate into a new fruit body, as because there is germination of a long pollen tube or hyphae, germination of the hyphae, these tapering apical regions needs to be moderately thin. The epithermal layer should be moderately thin for the germination to occur. And another important characteristics of this conidia is like the germination is bipolar, which means it can germinate from both the apical regions. Now, another important thing is that you can see in the image a dark colored circle. A dark colored circle, this is actually the sexual stage of this pathogen, Cochlea bolus miabeanus. In this dark circle, as you can see among this is known as the perithecial fruit body of the sexual stage of the fungus. The fungus, when going through the sexual stage, forms into a perithecial fruit body. Now, what is a perithecium? If you ask me, see, a perithecium is a hollow fruiting body which is round, spherical, and microscopic, obviously. And now this perithecium contains asci or ascospores, which are the sexual spores which reproduce to form a new fruiting body. Now see, if I give you through a more closer look of these perithecial fruit body, you can see it contains a neck and a long beak-like neck. These are, the layer is pseudo parenchymatous layer, the skin, the outer epiperidium, and the neck contains spine-like, thorn-like teeth or something which are present inside the ostiole. Now the ostiole is the opening through which you can see these long, slender looking ascospores present inside the perithecium, which are disseminated from this ostiole. Now let's learn about the ascospores, the meiotic spores. Now these spores are also segmented like the conidias, but the segmentation, the septate regions are a bit different from the ascospores, from the conidiophores. These ascospores have a different type of pattern of segmentation, and that's what helps us differentiate between an ascospore and a conidiophore if they are already given. After learning about the causal organism, now let's learn about the disease cycle of the causal organism or the pathogen, how the causal organism continues its life cycle parallelly along with the spreading of the disease inside the host. Now, before learning the disease cycle, we must understand this pathogen actually is a localized disease. The brown spot disease is a localized disease. It cannot spread very easily, and it actually remains stagnant in the areas where it infects itself. They build up inoculum from the infected region and disseminates further by the production of conidiophores, which disseminate conidia into different regions for further infection. Now, this disease spreads from one plant to the other plant by two ways. One is from seed. From the infected seeds, as I have shown in the identification, that how the seeds get infected and how the infected seeds look like. And second, from infected plant debris. Now, from the seeds, we know when the infected seeds are sown in the soil, they do not hamper the growth of the seedling. The seed grows normally, but the infection remains. Now, when the seed is growing, the seedling develops and the coleoptile gets infected, and then gradually it affects the seedling, showing gradually when the seedling grows, the blighting symptoms appear in the different leaves, different parts of the plant. Now, the infected regions of the plant, like the coleoptile, the leaves, etc., produce a large number of conidia. So, first of all, what happens? The conidiophore comes out through the stomata. The green parts obviously comes out of the stomata and develop conidia. Okay. Now, what happens? This conidia, how does it infect? This conidia infects the inflorescence. Now, the infected inflorescence, obviously this is the part where the infection spreads. Now, these inflorescence contain the seeds. Obviously the seeds infected. Now, from these it germinates. The germinated conidia as shown how the conidia germinates and activates the mycelium, which in turn host infection, causes the overall host infection, all parts. Now these again go on continuing this. Now this is the secondary cycle. This is the primary cycle. This is the primary cycle. This continues one season after the other. This is the primary cycle. Now, as I said, the secondary cycle is which continues, repeats in a single season many times to increase the progression of this infection inside the crop or the plant or the host. Conidiophores release conidia from conidia as it infects the host infection, spreads, new infection, and again conidiophores. So this is the secondary cycle, and this is the primary cycle. So this altogether is the disease cycle of brown spot disease of rice. These occurs by the seeds. Now when this occurs by the seeds as shown, and when the infected plant debris, they occur through a different type of primary cycle, all the same. Like this is the seeds, but when there are infected plant debris, as we know, all parts of the infection spreads, and these parts, when remain infected, they are not removed after the crop is harvested. They spread to the conidiophore to again a new plant, and the disease goes on spreading from one plant to the other. Now this is the whole disease cycle. Now let's learn lastly, the last important thing, last factor of a disease are the environmental factors, and how we are gonna treat this plant, infected plant, to stop the disease and stop further infection. Now let's learn about the environmental factors and the methods of treatment. Now we have reached the last part of the topic that we need to learn about the brown spot disease of rice, is about the environmental factors. The third factor of a disease is the environmental factors, which actually benefit the causal organism to grow and spread the disease in the host, or it may even, conditions may be unfavorable for the fungi or the pathogen to spread. So let's know about what are the predisposing factors or the environmental conditions which actually favor or inhibit the disease to spread. So first learn about the predisposing factors. So now the disposing factors are the factors which favor the development of the disease. So first is a relative humidity. If the environment is humid of about 92, this is an average value, 92.5% or above, favors production of conidiophores. Second, we said about humidity, it can even be a cloudy weather devoid of sunlight. Sunlight is something which can hamper the growth of any type of fungi, because any fungus requires a very wet and a humid condition as well as warmth. Not a very scorching sunlight, but a warmth and humid condition to grow, and higher day temperature. So both of these are required. Around 29 degrees Celsius in the growing season. Favor the rapid growth and places where there is shade. If any part of the plant is devoid of direct or diffuse sunlight, maybe in a shady condition, with high humidity area, humid condition. This can also favor the growth of fungi. So we need to keep in mind these three conditions should not be present in a area where we are planting the seed. So these can favor the growth of fungi. Even if the seed is infected, the plant is infected, if these conditions are unfavorable, not present, not available for the fungi to grow, the fungi may even stop growing, and initially it will die and not spread inside the plant. So after this, what needs to be learned is the disease management. How to manage the disease, how to treat the disease, how to stop the disease. That is called disease management. Now, disease management has a variety of methods. It can be number of methods we follow while culturing the rice plant. There may be methods which we can do while harvesting the plants, harvesting the seeds of rice, and even all throughout the growing season there may be even many methods which can actually be the use of different chemicals which can be spread all through the growing season, like fungicides and different things which can actually inhibit the growth of this fungi. So I'll be discussing in brief about some, like cultural methods are sanitation, sanitizing, cleaning and burning the previous year's infected plant debris are effective to reduce. So sanitization of the remains of the plant debris or removal of collateral hosts. This is important because see, when it is not the season for the rice to grow, the fungi may be present inside the growth area, maybe inside the field, in the collateral hosts, and may be growing, waiting for the time when the crops are sown and to spread into the primary host, which it really survives. So if we remove those collateral hosts, a high amount of infection can be removed. And lastly, irrigation of water. Enough amount of irrigation of water is required for removal of the infected areas from the field without any disease. And then leaching of metals. Leaching down the metallic elements like iron, manganese, potassium from the soil during rain should be checked. Like leaching down the metallic elements like iron, manganese and potassium from the soil during the rain. Otherwise the plants can be more susceptible to disease like manganese, iron, potassium. So these metals need to be prevented from leaching inside the soil. Now, usage of nitrogen fertilizer. Optimum nitrogen fertilizer actually has proved to be an important weapon for the reduction of this disease. So, application of nitrogen fertilizer can also stop the disease from spreading. Now, there are many physical methods like growing the crop in high sunlighted area area with bright sunlight, a field which has sunlight for more than 6 to 8 hours, and even treatment with hot water. These are the two things, physical methods. And lastly, as I said you, chemical methods can be dry dressing of the areas of the infected places of the plant, and foliage treatment. Foliar fungicides can be spread on the leaves. So these are the methods which we can use for treatment of the disease.